Protein purification is a laboratory technique used to isolate target protein in a complex mixture. Scientists use this technique to study protein function and structure, drug production, and industrial application because it can obtain highly pure form of desired protein while removing contaminant efficiently. Before going into more details, let's look at the general step of protein purification. First is sample preparation. We extract protein from biological sources such as yeast or bacteria and then disrupt the cell to get the protein sample out. After that, we perform initial separation and concentration by removing unwanted components and concentrating the sample. Following that, we're using specialized chromatography method to refine the separation for purification. Finally, we can evaluate the purity and quality of the isolated protein by various purity and lysis techniques. We say using specialized chromatography methods for purification, but what are those chromatography methods we can use and why is the difference? First is affinity chromatography. This method separates protein based on specific binding to a lesion. In a simple word, prepare binding site only for target protein after the protein bind to the site and release when no more other components. Then we can get high purity isolation of target protein. Second is size exclusion chromatography. This method separates protein by size. Large protein will go through the porous bay faster than small protein. This method can be used for protein size separation, removal of aggregate or buffer exchange. Third is ion exchange chromatography. This method separates protein based on their net charge at a given pH. It is useful when we need to separate protein have a similar size but different charge. Fourth is hydrophobic interaction chromatography. This method is carried out by adding the protein sample to a high salt environment which enhance the hydrophobic characteristic of the protein, allowing it to bind to the hydrophobic ligands on the chromatography column and separate from the mixture. As the salt concentration decrease, the hydrophobic interaction weaken and enabling the protein to be eluted. And the last one is chromatofocusing. This method separates protein based on their isoelectric point using the pH gradient. The isoelectric point is a pH at which a protein has not net charge. Each protein has a different isoelectric point, so as the sample passes through the chromatography column and the pH gradually decreases, each protein will reach its specific isoelectric point. At the point the protein has not net charge and no longer interact with the column, allowing it to elute separately. After purification step, we have to assess the purity and activity of protein to ensure the protein success. Therefore, how can the purity and activity of protein are assessed?
Next, I will be discussing the key techniques used to assess the purity and activities of protein during the purification process, which are critical for ensuring that our proteins are both pure and functional, particularly in fields like biopharmaceuticals. So, let's begin with purity assessment. First of all, there's the SDS page, which is a powerful technique for separating protein based on their size. This method provides valuable insights into both the purity and molecular weight of our samples. Following SDS pH, Western blotting can be used to confirm the presence of specific protein. This technique utilizes antibodies to verify the identity and purity of target protein. Besides, another key method is ELISA. ELISA uses antibodies to detect and quantify specific protein with high sensitivity. This technique is especially useful for identifying host cell protein in biopharmaceuticals. Next, let's move to activity assessment, which evaluates whether our protein retain their functional capability. First is the ELISA. ELISA also plays a role here by measuring protein activity through interaction with antibodies or ligands. Furthermore, the radioligand assay are another method which utilize radioactively labeled ligands to study protein-ligand interactions. By measuring radioactivity, we gain insight into the binding behavior of protein. Last but not least is the surface plasmon resonance (SPR), which provides us real-time data on protein-ligand binding interaction. This label-free method offers kinetic data, giving us a deeper understanding of dynamic of this interaction.
Purified proteins have vast application in both research and industry, playing a key role in advancing science and improving quality of life. The examples application of using protein purification techniques in research such as structural biology, disease mechanism research, and protein-protein interaction studies. First of all, for structural biology, purified proteins are indispensable for studying their three-dimensional structures. Techniques such as X-ray crystallography and MR spectroscopy and also cryo-electron microscopy provide insights into protein architecture, aiding in drug design and molecular biology research. Besides, in studying diseases such as neurodegenerative disorders or cancers, purified proteins reach allow researchers to model disease in vitro and also investigate how protein misfolding or aggregation contributes to disease progression. To understanding protein interactions is vital in biology. Thus, purified protein, which used in co-immunoprecipitation, surface plasmon resonance (SPR), and also E2 hybrid screening, reveal how proteins interact and which is crucial for deciphering cellular pathway. For industry applications, biopharmaceutical production, diagnostics, and food and beverage industry are the examples of using protein purification techniques. First of all, purified therapeutic proteins are essential in medicine such as insulin, which is using for diabetes treatment, growth factors using for cell therapy, and also monoclonal antibodies for targeted therapies. Besides, purified proteins are integral in diagnostic tools for detecting disease such as HIV, COVID-19, and also tuberculosis, as well as form the backbone of immunoassays such as ELISA and also Western blotting. In food and beverage industry, purified proteins are utilized for enhanced texture in products, improve nutritional profiles, and also aid in food preservation via an enzymatic process. After discussing so much about this technique, which is purified protein, but what is the limitation that we will face when we carry out this technique? So the first one is protein stability. Due to the complexity of artificially designed non-natural proteins, they are prone to issues such as precipitation and reduced quality during conventional purification process. Second one is cost and time. Some purification methods can be costly and time-consuming, which limiting their practically, especially for low abundance or labor proteins. The acquisition of high-purity target proteins requires the cooperation of multiple devices, which increase the research cost. And the last one is scaling up of purification processes. It because techniques that work well at the laboratory scale may not be easily scalable for industrial production, posing challenges for large-scale protein purification. So example of challenges that faced before by using this technique is challenges in the purification of recombinant tech proteins by affinity chromatography. The cases is like facing the ligand availability and cost, which specific affinity ligands may not be available for all target proteins, and custom development can be expensive. When considering the therapeutic proteins, the use of affinity pairs is not the preferred choice due to the need to strictly control the presence of the tag in the final product. Moreover, the additional steps of tag approval and further purification contribute to higher associated costs and low yield.
As a conclusion, protein purification is a cornerstone of both research and industrial application, enabling the isolation of specific proteins from complex mixtures. The diversity of available methods such as affinity chromatography, ion exchange chromatography, size exclusion chromatography, and hydrophobic interaction chromatography ensures that purification can be tailored to the unique properties of each target protein. Techniques like SDS-PAGE, ELISA, and spectroscopic methods further support the assessment of protein purity and activities. Despite the effectiveness, protein purification process faces several challenges including protein stability, high costs, and scalability issues. For instance, the production of therapeutic proteins demands stringent controls to remove tags and impurity, adding complexity and cost. Moreover, methods that perform well at laboratory scales often struggle with industrial scale implementation. Overall, continuous advancement in purification technologies aim to address these challenges, offering more efficient, scalable, and cost-effective solutions. The integration of these technologies remains vital for advancing structural biology, drug discovery, diagnostics, and industrial production. That's all from our group presentation. Thank you.